So I'd like to speak about the stability principle for information extraction from data. So this is something me and my group have been working on for the last 5 years. I want to start with this, that's what got me started on this, is this article by two editors of immunology journal called Infection and Immunity. And they basically said that for the 10 years before that, there were a lot of retraction from PubMed, and many are so-called errors, and other are called fraud. And you can guess a lot to do how they did the statistical analysis. And I would like to say that push forward this stability principle is really building on what people have done in statistics and other people try to make it called the principle so it gets more attention and get more applications of it. So I'll say that statistical stability is a minimum requirement for scientific reproducibility, will really help reduce the positive rate and it's easy to use. But there are two important components in this our core principle application. You need to have a reasonable data perturbation scheme for ID data like bootstrap and cross-validation, all like eligible forms. And you also need a stability measure and relative to what. So the task has to be on the scope, like you want to do estimation, prediction, or model selection. They will lead to different results. So I want to start this. This is also a motivating example why I started working on this. Is I've been working with Jack Lance's lab in Berkeley on neuroscience problems. And we have data for fMRI, which is non-invasive and good coverage in direct recording of neural signals. And each voxel, I'm going to use this again, is a 1 millimeter cube, which contains hundreds and thousands of neurons. So the data comes as a spatial time series. Each voxel, we have a time series. And the input are natural movies I'll show you. So they got quite a bit coverage about 5 years ago. So on the right, your left-hand side is the movie a subject was watching, a human, and this is our reconstruction. So it got into Time magazine for one of the best 50 inventions of the year. But the question is, this is a prediction, this is good engineering, this is some validation to the model. Can we do some science? Do we learn anything about how the brain processes these images? So, question? A key step in this movie reconstruction, we develop a forward model with a lot of neuroscience knowledge, V1 and V2 processing, Gabor-like, and we have features which are located at different locations on the image or frame. And then we did something like a lasso fitting to predict the neuron signals. And each voxel, we had one model. But the science is not about you can reconstruct a movie, it's about which voxel is driving by what stimuli. It's not conclusive, it's suggestive, but can we say anything about which stimuli might be driving which voxels? So if you look at the lasso plus cross-validation selection of features, it happens that I can locate my features to a position on the image. You can see that for the three very good voxels, we have correlation prediction score about 70%. We use correlation because we cannot predict the magnitude in brain. It's all of the map. So you know that it's not quite right because we know that voxels, even they have hundreds and thousands of voxels, they don't care about the whole image. But for prediction, they do a very good job. So suspicion is that because of correlation. So then we wanted to apply stability principle. So the question is which perturbation scheme to use and what stability metric we're going to use. So let's take a step back. So data perturbation is not really a new idea. If you go back to the 50s, and Tukey actually said that, Huber said that Tukey really preferred to rely on the actual batch of data at hand rather than a hypothesized underlying population, which it might be a sample. So Tukey, of course, wrote this paper called Future of Data Analysis in 1962. It's really one of the early papers on data science. So if you're interested, you should take a look. And there's a long history about different versions of data perturbation. This goes to the 40s. Jacknife, subsampling, cross-validation, and bootstrap. They're all very useful for most cases when you have kind of symmetric ID independent data. And the question is for dependent data. That's, I think, is an open question. And I want to make a case that even for the very cornerstone of classical statistics, which is central limit theorem, is actually a stability result. I don't know how you learned your central limit theorem. I didn't learn it this way. So Terence Tao's website has 7 proofs of central limit theorem. One particular one I think is very related to my talk is called Lindenberg swapping trick. So you have normalized sum of ID random variables. You can swap in another random variable one at a time with the same mean and variance with some control on higher moments. And with some smoothness assumption, you can show that then the limiting law doesn't change. This is really a stability result. And you can show that the limiting law satisfies ODE and the unique solution is normal distribution. So this is that really what drives central limit theorem is this stability result. Modern times, there's random matrix result. If you want to generalize from Gaussian case to non-Gaussian case, you can use this swapping trick. And for percolation result, you can use this swap, like stability kind of argument. And in our own work with my colleagues, Crowe and Bickel and Lim and Bin, we show that for high dimensional M estimation, you have somehow stability kind of argument to lead to limiting result. None of these are central limit theorem. They are limiting result. So this is really saying that stability is more essential than any limiting result. And you look at some concentration results, you have kind of stability conditions, and there's work from as early as the 70s in kind of statistical machine learning literature that people show that stability is closely related to good generalization performance. So I'm just saying that this is a very fundamental concept and it's very related to the big data we can do easily, and therefore I want to flesh it out and make it like, call it a principle. And even you go to the 90s in the like AIC, BIC era, that people use bootstrap to do model selection, like Bryman and Shaw's work. And more recent time in the Lasso literature, sparse modeling, people did different version stability consideration and derive different things. So what we do here, we take the perturbation from cross-validation. The reason for Lasso, people use GlimNet a lot. People already using cross-validation, so I don't want to introduce a random split. I want to use cross-validation or perturbation, but we design a new metric for estimation. So cross-validation is really, you want a stable result for prediction. So what we have here is we want to estimate the regression function. For this case, it's kind of have linear form, x beta. We do the cross-validation, say 10-fold. We have 10 beta estimator. And we look at the variance, the upstairs, relative to the central mean, the grand mean. You have all the 10 estimates add together and cook up regression estimator, x beta hat bar, and you look at the variability. And then you compare with the grand mean estimate. This actually has a statistical interpretation. It's really the reciprocal of a test statistic for testing the regression function being 0. So for each fixed lambda. So what you're doing is that you look at this test statistic for fixed lambda, you're looking for the most significant on the path testing this hypothesis. And that's what I'm going to want. Because so I care about regression. I don't care as much as the prediction. But however, if you think about that, if you really go for stability, constant is very, very stable, right? So it's not enough. You need to pack to something that fits the data. That's why we go back to cross-validation. We said, we like the cross-validation measure on how well the model fits the data. But we want a smaller model, which is more stable. So we take the lambda that's selected by cross-validation, and then we choose a bigger lambda, because we want a smaller model, that have the local minimum of my design, the new estimation stability criteria. And that's my ESCV choice, estimation stability with cross-validation. So it's really using cross-validation for model fitting, but same time, look for stability with a new metric. And that's what you get. So for that problem, when it's all over the place, you really trim down to something a lot more sensible. Okay, so this, still there are a lot of false positives here, but it's much, much better than all over the place. It just biologically makes a lot more sense. And I thought we lose a tiny bit for these 3 voxels in terms of prediction error, but for over thousands of voxels we have, we barely lost on average 1.3% in terms of prediction error, because we have each voxel has a model. This is like 3,000 voxels. And in terms of the model size, we reduced on average by 60%. So this is actually, and we thought we're going to lose prediction, but we're willing to lose prediction error for the sake of interpretation. But in this case, actually, we don't lose much at all. We got good on both words. And if you think about prediction error captures bias and variance, and stability is more related to variance. So together, these two quantities both capture both the variance and bias. Actually, I'm writing a textbook with a student. We hope to use that as the main theme to tie it together, statistics. So I want to show you case 2. So I talked about this one version of data perturbation. But often we also make choices about what model to use, what method to use. So for clustering, you can do hierarchical clustering, you can do k-means. And for regression here, you can do ridge regression, you can do lasso. Actually, we have another project asking which one do you interpret with. So there's a need also to ask for stability relative to method choices or model choices. And so we've been working on, that's another success story. We use this stability principle. We've been working on genomic project. It's more recent than the movie project. We have a non-convex optimization problem of dictionary learning or non-negative matrix factorization. And the question is, how many components we want in the dictionary? For us, that means how many pre-organ regions for the Drosophila developing embryo. So if you think about non-convex optimization, really, each of the non-convex optimization actually have multiple models. Each mode is a well-defined model. They just happen to share the same parametrization, which is great for computation. You can move easily from one to the other. So when you look for the different modes, try to bring stability in, you basically want your result to be relatively stable to the modes or different models represented at the mode. So that's why I call this as a stability relative to model perturbation. So here, we cannot go for that estimation stability anymore because we have permutation variance of the problem, and we went for Amari index using correlation matching of two matrices. Right? So that has to be specific to the problem. So here's the problem, and the paper just appeared in PNAS. I'm a co-lead on the data side, and my biology co-lead is Irvin Fries from IBL. We made it to IBM News and got picked up by a lot of the online science news. So the writer did a very good job. It's really mapping a cell's destiny. That's the rendering of the first author, Siqi Wu, who is really a major force for this project. We really want to know, when you look at embryo genomic images, where these different cells will form into the future organs. So that's the scientific problem. So here's the data we're looking at. We have each image is a gene. We know the intensity, relative intensity of each gene, and where they express. So the design principle is association by guilt. If two genes locally co-vary, they probably interact. They're doing something together. There are high resolution data I want to talk about. So that's microscope. It's high throughput. You find them, and you put them into the same view, and then you put them together. So there's a lot of pre-processing. Actually, we use everything's pre-processing, some pipeline to do this. So after all the processing, which is very, very important, 80% of the work, but we become like 405 big pixels. So the different stripes, you can see, genes don't act everywhere. They do different things. So you think about genes as workers. They work together to build different rooms for the house, and the house is the embryo. Okay, so they team up differently to build different organs. This is pre-organ time. And proof of concept why genes have different functionalities at different locations is the famous gap gene network Nobel Prize winning work by this German scientist. You look at giant, it's one gene, giant, same gene, appear in two locations. So the giant gene, which means you knock it out, the embryo becomes giant, does different things at two different locations, at the anterior and at the posterior. So this is well known. This is a very famous gene network that's really decide how the vertical segmentation happens for Drosophila and similarly for human embryo. Drosophila and us share 75% of the genes. Okay, so we're not that different. We can learn a lot from Drosophila. So we borrow actually from neuroscience to do this decomposition. So the concept of building block came from science, saying that we know genes do different things in different locations. And how do you find these meaningful blocks on the image or regions? So we end up using non-negative matrix factorization. We line up lay out all the images into a vector, and we're looking for a dictionary, and we call the columns, when you put it back into image format, principal patterns. And you have coefficients. Okay? So the question is how to choose K. How many regions we want this dictionary to have. And it turns out, that's what will show you, that the regions turned out to be completely coherent. We didn't put any spatial constraint during this non-negative matrix factorization, but the principal patterns, as shown, PP1, PP2, PPK, come out to be biologically meaningful and contiguous. It's because there is this underlying process, because it's spatial, the organ is spatially continuous. And then the genes have to work together to respect that boundary to make things happen. So this is an indirect proof that we're kind of on the right track. So the new contribution, besides having the idea using sparse coding or non-negative matrix factorization here, is to use stability to select the number patterns. And we design, so you have random starting point with, say, 21 randomly selected images from the data as the beginning D, and you alternate. Then you start again with another 21 randomly selected images. You do it again, and you match them by correlation, because D1, D2, D3 doesn't have a meaning. It's collectively, they have a meaning. So you match the best D from another run, and you do 1 minus of that correlation, and that's your dissimilarity. And we find the most stable K is 21. And we did look into 22 because we also want to do some stability analysis to make sure we didn't miss anything. 22 is you split one of the 21 patterns into 2. Okay, so we did a lot of follow-up analysis to make sure any decision we made is not driving the conclusion. So that's kind of high-level stability, decision-making stability. So that's what we ended up with, 21 principal patterns. And you can see a group of vertical ones, a lot to do with gap gene network, and horizontal ones, posterior, anterior. So when we show it to biologists, they were really, really happy. They think it makes sense to them. But that's not enough, right? That's the first step. And we want to, you can lay out this famous, in the middle is a fate map. It's by old technology of laser ablation. They figure out at this stage, this purple area will develop into more ventral neurogenic regions. So now we lay our decomposition on top of that. And if we know, say, a particular gene is heavily expressed in PP4, we can already have conjectured functionality, saying this gene probably has something to do with neuron development. Okay? So by combining the new technology with the old fate map, we already have conjecture functionality. And 25 of the genes we're looking at are so-called computed genes. They know they're genes because they bind in the right way, but we don't know their functionalities. So this gives a lot of congested or suggested functionalities by see where they express heavily and map back to the fate map, because we know the fate map later what they will develop to. So we have a website. Other biologists can go there and try their favorite genes and see what happens. So that's one validation. And now we want to reconstruct the gap gene network. We set it up as a benchmark. Can we reconstruct the gap gene network? And we have to have a locality. So what we did, we used all these principal patterns as a mask so that we only look at those regions and look at correlations. Sometimes we put 2 together because for repression, you have to put 2 together to know they repress each other. So sometimes PP, sometimes 2 PPs together, and you have this correlation network or local correlation network. Lots of work. So the paper is 6 pages. We had 40 pages of supplement material. So a lot of details in there. And this project from the first paper, 4 years in making. So we reconstructed 11 links, 10 out of 11 links. There's a lot of work why that link, GT to KL, which local gene network I should look for that link. So it's a lot of details hidden under this slide, but it's in the supplement material. You can take a look. So we're pretty happy that this is another validation that what we did seemed to be working, but we still want more. We want to do experiment. So we went for CRISPR-Cas9 as a gene editing technique, which if you haven't heard about it, you should be, because it could alter the human race forever if that ever get, now people are saying that we shouldn't work on human or not the stem cell type genes. You can do local genes, we won't get into the gene pool. So there's a fight between Berkeley and MIT right now. Jennifer is here in Berkeley, and she and her collaborator, Computer, got the breakthrough prize 2 years ago, $3 million each. So this is the hottest thing in genetics. It's a very easy, relatively easy way of editing genes. So it took a while for this to work, because most of the people have done not in the living organism, and Sue's lab is one of the first to do that in living organism, and things is harder. So here's the result, early result. So we knocked out this computational gene called CGX, and one well-known gene is one of the Peru genes about even skip, like where you lay the even strip and the out strip, so controlled by 2 genes. You can see that for this image, that seems the head got bigger. Okay, but that's not something you publish. But we know this is very important. This is 2 or 3 hours after hatching. 16 hours, the embryos all die. So we know it's a lethal gene, but that's not enough. You have to say why and how the computation will work. We have some idea about what might happen, and we got more genes, so that's more like a small sample of genes. So the color, the red is the mutants, and the green is the white. So you can see there's a shift on average. We're running more experiments. Seem to be bigger, but it's also bigger variability. So what happens is that when you knock out some gene, the whole system gets off balance. And it's not always, it's also increase of variability. It could go both ways. So it took a long time for us to figure out how to measure it, because different embryos have different sizes, why things are comparable. So we're doing more experiments. So here's a summary. I think stability is a minimal requirement for scientific reproducibility. I believe it's important to bring it up as a principle so people can use it more and something to talk about. It's intuitive and easy to use. And we have shown success in 2 cases. And we have shown with random forest, with also good results, and also with some advanced light source data from IBL, also a good result. And ongoing and open problems, we don't have theory really for ESCV and stability-driven non-negative matrix factorization. And the dependent case is what we're working on, but just hope other people will join in this understanding and using the stability principle. So here's a team for neuroscience. You all did a lot of the stability stuff. And we have also a team for the Drosophila project, and 3, like my group, and IBL biology, and Tsinghua CS group, because we're also building software on Spark and Fiji so that other people can use it. And here are the 3 papers I took material from. Thank you.